This is the fourth part of the video lecture on quantitative bioanalysis. In this presentation I will give a brief overview of the process of drug development and the role of bioanalysis in this field. And in addition I will focus on a practical example that I hope will give a good illustration of how quantitative bioanalysis is used to answer all kinds of clinical questions. Drug development is long and expensive. Out of every 10,000 drug candidates, only one will finally make it to the pharmacy in a process that takes about 12 years and costs close to 2.5 billion euros. After testing in animals in the preclinical phase, a potential new drug is given to healthy human volunteers in phase 1 and finally tested in the intended patient population in phases 2 and 3. The clinical research with patients is typically performed in a hospital setting, while phase 1 clinical research in healthy volunteers is done in specialized units. In all phases of drug development, but especially in phase 1, bioanalysis is an essential part of the work. Among other things, the drug concentrations that are generated are used to establish pharmacokinetic parameters for the drug, and the levels of endogenous analytes, such as biomarkers, help evaluate how effective the drug is. Routine measurements, for example common biomarkers, are usually performed in hospital labs. More specialized measurements, such as the concentrations of the new drug, are typically done in contract laboratories. I now want to show you a practical example, and for that I get back to risperidone, one of the most widely used drugs against schizophrenia. At PRA's bioanalytical lab we have been involved in a large part of the development of this drug. It has been on the market for over 20 years and it's given either orally, every day, or injected every 2 weeks. Important for the proper bioanalytical support is the fact that risperidone is metabolized by the cytochrome P450 isoenzyme enzyme CYP2D6, and hydroxylation leads to the formation of 9-hydroxyrisperidone, which is also pharmacologically active. The metabolite is also a drug itself against schizophrenia, and it's called paliperidone. One of the first things to do when setting up a method is to evaluate the molecular properties of the analyte. Here you can see some important characteristics for risperidone and its metabolite. The relatively high log P values indicate that the analytes are non-polar, and the basic pKa value of around 9 shows that they are charged at physiological pH values. We will use this knowledge to develop a proper method. I will show you how we did the bioanalysis to support a clinical study in which the differences in metabolism of risperidone was investigated. As you may know, most humans are extensive metabolizers for CYP2D6, which means that this enzyme has a rather high activity and will metabolize a large part of risperidone to the 9-hydroxy metabolite. About 10% of the population are intermediate metabolizers and they have a lower enzyme activity, while another 10% are poor metabolizers who will metabolize almost no risperidone. And finally, a very small percentage are so-called ultra-rapid metabolizers who have a very high CYP2D6 activity. We had to determine the concentrations of risperidone and 9-hydroxyrisperidone in plasma of extensive and poor CYP2D6 metabolizers after a single oral dose of 2 mg of the drug. Requirements were that we had to use no more than 500 microliters of human EDTA plasma per analysis, that we had to go down to 0.1 nanogram per ml for both analytes, and of course that the method had to be validated according to international scientific guidelines. Our road from sample to result was as follows. We used SPE to extract the analytes from plasma, we used HPLC for separation, and MSMS for detection. Well, let's start at the end. As a reminder, with MSMS detection, the analyte that elutes off the HPLC column is ionized, and in the first quadruple, ions with one specific mass-to-charge ratio are selected. For risperidone, this is 411.2. All ions with other mass-to-charge ratios are removed. In the second quadruple, the selected ions are fragmented, and in the third quadruple, a fragment ion that is specific for the analyte is selected. In this case, its mass-to-charge ratio is 191.0. These ions are then finally detected. Risperidone has a mass of 410.2 and it picks up a proton and forms an ion with a mass of 411.2 and a charge of 1. As you can see in this figure, it is fragmented almost exclusively to an ion corresponding to the right-hand side of the molecule, which has a mass-to-charge ratio of 191.0. For the internal standard, something similar happens. Because of its 2 13C atoms and 3 deuterium atoms, it has a mass 5 units higher than risperidone. The mass-to-charge ratio after the ionization is therefore 416.2. And after fragmentation, only 2 of the 13C atoms are left. So the fragment is 2 mass units higher than the fragment of risperidone at 193.0. For risperidone, for 9-hydroxyrisperidone and its internal standard, we found comparable results. And here is a summary of the applicable mass-to-charge ratios for all 4 compounds for the first and the third quadrupole. As you know, HPLC is a technique that can separate mixtures of molecules by pumping a mobile phase through a column filled with a stationary phase. For our risperidone method, separation was very important because there is a minor metabolite, 7-hydroxyrisperidone, that has the same mass as the 9-hydroxy metabolite. It has exactly the same Q1 and Q3 mass-to-charge ratios, so these 2 metabolites cannot be distinguished by mass spectrometry. Therefore, an HPLC separation is essential if we want to obtain accurate results. Without a separation, we would of course find a sum of 9-hydroxy and 7-hydroxy, which is not correct. Fortunately, we could easily find a good combination of stationary and mobile phases that allowed separation of the 7 and 9-hydroxy metabolites, as you can see in the right-hand chromatogram. And as expected for risperidone, we see no interference. To extract the analytes from plasma, we used solid phase extraction. The sample is applied to a cartridge filled with a solid phase, to which the analytes bind and from which as much as possible of the matrix is removed by a rinsing step. Finally, the analytes are collected by eluting them from the solid phase. Here you see the charge of both risperidone and 9-hydroxyrisperidone as a function of sample pH. At the pH of above 10, all risperidone molecules are neutral. At low pH, below around 7, they are all positively charged. We can use this to extract the analytes by a negatively charged SPE phase or cation exchange phase, which attracts the positively charged analyte ions. So, the extraction steps are: adjust the plasma pH to below 7, in this case we used 6, to trap the analytes. Wash the cartridge at low pH, in this case 4.5, to remove matrix compounds. And then elute the analytes at a pH of above 11. At this pH they are neutralized and will be released from the SPE phase. Both analytes had a high recovery of around 95%. And to be complete, here is a detailed extraction protocol that shows you the different steps. Conditioning of the cartridge, loading of the sample, washing, and dilution. In this way, a good method was obtained with results for accuracy and precision that met the international criteria. Also, the stability of both analytes was found to be okay. The method was used to determine the risperidone and 9-hydroxyrisperidone concentrations in plasma from extensive and poor metabolizers. Example concentration time curves are shown here. As you can see, for an extensive metabolizer on the left-hand side, the metabolite concentrations are higher than those of the dosed drug, because risperidone is readily metabolized to the 9-hydroxy metabolite. For a poor metabolizer, on the right-hand side, only very little risperidone is metabolized. Interestingly, the sum of both analytes is about equal for poor and extensive metabolizers, which means that the anti-schizophrenic effect is similar for both groups of patients. In conclusion, I hope that this example has demonstrated that drug development needs good quantitative bioanalytical support, and that suitable methods can be developed by proper optimization of extraction, separation and detection. And with that, we are at the end of this lecture. But please have a look at the video in Dutch that gives you a feeling of how quantitative bioanalysis is used from day to day in a contract research laboratory. Thank you for your attention.